Okay, so I guess I'll launch into part 3. This is my last talk, and probably the part that I've, is closest to home in many ways. I am a neuroscientist. I only masquerade as a, data scientist, and so I spend most of my time thinking about the human brain. So today, what I thought I would do is just talk to you about kind of two different realms of applying gene co-expression methods to the human brain. First realm being in the adult brain, second realm being in prenatal or fetal human brain. I should say that everything, hopefully you can see the, ability of what I'm going to tell you to generalize to other biological systems. So I'm a neuroscientist. I became a neuroscientist to study the human brain, and one of the biggest elephants in the room in neuroscience is that we don't really have a parts list for the human brain, right? I mean, we know what the major cell classes are, but there's no consensus on how many different cell types there are or how we define them. And so, this is one of the major goals of the NIH Brain Initiative that was announced by President Obama, is to try and, conduct a census of cell types in the brain. That is kind of one of the areas where my lab focuses a lot of its efforts. I can be of two minds about the state of neuroscientific research, which is why I have this slide. On the one hand, you see all these papers coming out in fancy journals that are so definitive in their titles, and this thing regulates this or is required for that and blah, blah, blah. And on the one hand, it's really impressive and unbelievable that we can do this stuff. But then on the other hand, you often feel like one of these soldiers trying to put Humpty Dumpty back together again. How do you integrate all of these reductionist gene, by gene studies into some kind of coherent narrative, particularly in the human brain, which is a mishmash of all these different cell types. You take a step back and you say, well, what makes one cell type different from the next is ultimately the genes that it expresses, right? In principle, they're supposed to have the same genomes, even if there are somatic mutations once in a while. So if gene expression really distinguishes one cell type from the next, and we have great technologies for measuring gene expression, then to me this seems like the most logical point of entry for trying to characterize cellular heterogeneity. Of course, it doesn't exclude the use of other criteria like morphology and position and connections and all of that, but those things are harder to measure, right? So it's thought, based upon some recent studies, that there are about 85 billion neurons in an adult human brain. And most of those are in the cerebellum. In fact, about 70 billion of those are in the cerebellum. Those are called granule neurons. There's an unknown number of glial cells, which are three kinds of glial cells primarily: astrocytes, oligodendrocytes, and microglia. How these things partition into subtypes is not entirely obvious, kind of poorly understood. There's also vascular cells, ependymal cells, choroid plexus cells, and who knows what else. So in my lab, we're trying to sort of synthesize all of these sort of pointillistic observations about individual genes into a cellular context, right? So when you see a paper about KCNC2, you know that that's a neuronal gene, right? Or BBOX1, we very much know as an astrocyte gene. So we're trying to use co-expression analysis to, study this problem of cellular heterogeneity in the human brain, and also how that heterogeneity arises during human brain development. And more recently, I've become interested in brain tumors, and particularly with my new position, I'm going to be spending a lot of time analyzing brain tumors, because in brain tumors, you have a new kind of cell type, which is a malignant cell type, right, which has been transformed by one or more mutations to either activate or repress a whole slew of genes that are not normally working together in the normal brain, right? So the same kinds of methods in principle, I think, will be very powerful sort of, identifying those. I had this slide earlier. I'll just kind of run through it again because in many ways it's the foundation of everything that I do. And, this thought experiment kind of occurred to me during my PhD here as a sort of reaction to what seemed to me to be a sort of, a limited way of thinking about the gene expression data that had been produced from human brain and also from any heterogeneous tissue, right? So it doesn't have to be brain, right? If you extract RNA from any tissue specimen that's a mix of different cell types, what are the odds that you're going to have exactly the same proportion of cell types across samples? 0, right? It's just not going to happen. And as that proportion or those proportions go up and down, it is inevitable that it will drive the co-expression of genes that are specifically expressed in a particular cell type, right? So to me, it's axiomatic, right? If you accept that different cell types express different genes and that their abundance will vary among samples, then this follows logically. You don't even need to do an experiment, right? At least it's enough for me. But of course, we had to do the experiments to prove it, and that's what I did in my PhD. This paper was actually the successor to a paper in PNAS that Steve mentioned earlier, comparing gene expression in human and chimpanzee brains. You know, I should say I was very fortunate in my PhD because I arrived with no background in programming or math really excessively, and landed in Dan's lab, who was already collaborating with Steve. And so, you know, I got to be an early adopter of WGCNA and work with the master on applying it for the first time to gene expression data sets from human brain. And the initial study, which was comparing 18 samples from 6 different brain regions in 3 humans and 3 chimpanzees, right? So a total of 18 and 18 samples, led to the PNAS paper. And for the most part, what we found was that there was a cerebellar module, there was a caudate nucleus module, there were some cortical modules, and some of them showed stronger preservation than others between human and chimp, which was interesting. But in many ways, the most important observation from that study was the identification of a white matter module, right? Which is not something you would have obviously expected when the study was designed. And I should say we didn't produce these data. They were produced by Svante Paabo's lab in Germany, and we just reanalyzed it. But it occurred to me in that analysis that, hey, you know, here we see this module. We see it in humans. We see it in chimps. It's got all these myelin-associated genes moving together. Clearly, that's telling you something about the proportion of oligodendrocytes in every sample, right? And again, it goes back to the thought experiment. There's going to be variation, and the oligodendrocytes have a very distinct transcriptome, right? So it's easy to pick out. And, but in principle, if it's true for one cell type, it should be true for all cell types, right, in principle. So that led me to delay graduating, which was, I think, incredulous. Dan was incredulous about that, but I didn't want to graduate because I had already started looking at all of these other data sets that had been coming out and seeing the same patterns over and over again in human brain. And so that led to this very sort of meticulous analysis of these 4 different data sets, which you know, the top one I cobbled together from 3 published studies, and it was 67 samples from different areas of adult human cerebral cortex. And at the time, I thought, wow, 67 samples, that's huge, you know, like, and at the time it was. The second data set was a different set of human cortical samples on a different microarray. So this is AFEU-133, this is AFEU-95, which is a very old platform. And then these 2 are from caudate nucleus and cerebellum on the AFEU-133 series. So we did, you know, diligent pre-processing, all the stuff I just talked about, and then ran WGCNA on each of the 4 data sets, identified co-expression modules which are labeled by colors, and where you see the same color recur in each of the 4 data sets, that represents a group of genes that had very significant overlap across the 4 data sets, right? So we were seeing the same groups again and again. When we cross-referenced the composition of these modules with experimentally validated markers of major cell classes, we saw that this M9, or the turquoise module, shown in, you know, all the 4 data sets, was very significantly enriched with markers of oligodendrocytes in each of the 4 data sets. This brown module with markers of astrocytes, and this blue module with neuronal markers, right? We also found modules that were related to microglia, subtypes of neurons, including inhibitory neurons, Purkinje neurons, and then other stuff that was not obviously cell type specific, like ribosomal modules, mitochondrial modules, and also a module that seemed to sort of track the amount of subventricular zone across samples from the caudate nucleus, which is kind of it's a neurogenic niche, right? So I don't want to give the impression, or there's 2 impressions I don't want to give. Number 1, I don't want you to conclude that every module corresponds specifically to a cell type, because it's most certainly false, right? Number 2, I don't want you to think that we know what every module means, because that's even more false, right? But what we can say from this is that there are these recurrent patterns. They show up again and again in these data, and they're completely invisible if you look at the data in terms of differential expression analysis. So why would you ignore them, right? So what do they look like? The turquoise module I mentioned, right? So here I'm showing the same 10 genes and the expression patterns of these genes in each of the 4 data sets, right? These genes include proteolipid lipoprotein, peripheral myelin protein, et cetera. And as you can see, they follow different patterns in every data set, but they're always correlated. Why? Well, because they track the proportion of oligodendrocytes across samples, right? So if you collapse this pattern to the module eigengene, we interpret that eigengene as a proxy for the relative representation of oligodendrocytes across all of the samples in that data set, which may vary for biological factors, biological reasons, or technical reasons like dissection artifacts and things like that. Here is a different set of genes. These are expressed specifically by astrocytes, right? So you can see among them, for the neuroscientists in the room, SLC1A3 is also known as GLAST1. SOX9 is kind of a famous astrocyte gene. So when you're working in a particular biological system, you begin to know the better markers of the cell types of interest, right? And when you see a module and one of the genes in the module with the highest KME is a famous marker for a particular cell type, pretty good chance that module relates to that cell type, right? And the enrichment p-values using a bigger set of genes will almost always back that up. Here's another set of 10 genes. This time, these are neuronal genes. You can see, I think there's one called synapsin-2, so neurons have synapses, so that's not entirely surprising. But again, through this principle of guilt by association, we implicate all of these genes sort of agnostically, right? Many of them have never been associated with these cell types in the literature. So this was 2006, 2007 when I was doing this, 2008 when this study was published. And since then, it's just been an explosion of giant data sets from human brain, right? Many of them from the Allen Institute, from the Nadia Stan's lab, and from other consortia. And so now it's just a totally different ball game, right? We can do things that were never possible before. So in my lab, we've been kind of systematically cranking through these data sets and trying to sort of map what are the patterns that we see and where do we see it. These are my 2 lab members who have been working on this. This is not even a complete list, and it's already up to almost 10,000 samples from human brain. And these are all non-pathological samples, right? So when you look at the distribution of these samples, we have pretty good coverage of the human brain transcriptome over developments. So GW stands for gestational weeks. So about a quarter of the samples are prenatal, right? And then a majority of them, probably about 2/3, are adult, so over 20, right? When you look at the distribution by brain region, you can see we have pretty good coverage of the whole human brain now, thanks again to the Allen Institute and others who have done these really, really heroic sampling strategies looking at trying to get total coverage of the brain. So that means I'm going to be analyzing these data for the rest of my life, right? Because when you think about the permutations, right, you can slice it up a gazillion ways. So we've been doing that. And one of the goals is to try and develop consensus signatures for major cell types in the human brain. And so we've divided the data set into anatomical regions, or I should say the data sets. So for a given region, we have between 1 and 9 independent data sets corresponding to between, 44 and 1,400 samples. And these represent all major microarray platforms as well as RNA-seq, right? So we're looking across everything. And we're building co-expression networks in each of these data sets. And we're trying to find modules that are enriched with known markers of a given cell type. And then we're calculating the KME values for all genes in the genome in that particular platform, and then integrating that across all the independent data sets to produce a consensus signature. So here's an example. Okay, so astrocytes. Here I'm showing all of the different human brain regions that we can now analyze. We'll start with one data set from cerebellum. This is a data set consisting of 54 samples from adult human cerebellum, all from different individuals. This is produced by the GTEx consortium, right? So here I'm showing you on the x-axis a number of modules which are labeled by colors, and on the y-axis is an enrichment score minus log p-value. And we're asking which module, if any, was significantly enriched with this set of genes that according to Ben Barres, are expressed much, much higher in astrocytes of the mouse brain than other cell types of the mouse brain. You can see they all hit this one module, right? And when you look at that module, so this is, each row is a gene, each column is a sample, and this is the eigengene across the 54 samples. And here are the top 15 genes in this data set ranked by KME, ranked by their correlation to this pattern, right? So again, based on this one data set, we would say at the RNA level, HEF right there is the optimal marker of astrocytes, right, based upon this one analysis. But it's just one data set, and we have many data sets. So let's look at another one. This is from frontal lobe. This is 44 samples, also an RNA-seq data set, but it's from a different source. It's from the Allen Institute. This time it's this black module that shows significant enrichment with this set of genes. When you look at the pattern, this is what it looks like. And let's see, there's a few famous astrocyte genes, like FGFR3 is pretty famous, but a lot of them are pretty much unknown. Here's a third data set. This is from midbrain, 182 samples. This is on a microarray. This is an adjuvant microarray. This is the module that shows enrichment with that gene set. Here are the top 15 genes in the module, right? So this is 3 out of about 60 regional data sets, right? For each of these 3 data sets, we can calculate the KME values for every gene in the genome with respect to every astrocyte module eigengene. And this is like the top 30 ranked by their average across the 3, right? And this is kind of just a simplification of how we do it. But you can see where it's going, right? We want to produce this consensus ranking where not across 3, but across all 60 data sets, where we find an astrocyte module, or a module that's very significantly enriched with markers of astrocytes, and we have multiple independent gene sets that mark astrocytes from different groups. We can produce this table, and then we can rank all genes in the genome based upon their predicted expression specificity for astrocytes, right? And this slide is a little busy, but it's showing you that ranking, one of the earlier versions of that ranking, where we're basically plotting that predicted expression specificity for astrocytes as a z-score. So genes that have a very high z-score are predicted to be the better markers of astrocytes, whereas things down here are not expected to be good markers at all. We've dropped lines at canonical markers. So for example, GLAST1, aquaporin-4, ALDH101, connexin 43, those are 3 canonical. I just realized you can't even see my mouse that I'm moving around. The red lines basically denote canonical markers of astrocytes. Oh, here we go. These guys, right? So those are canonical astrocyte markers that have high z-scores, whereas in blue, we're reporting canonical oligodendrocyte markers, and in black, canonical neuronal markers, right? And those do not have high z-scores, right? Because they're not specifically expressed by that cell type. We've done the same thing for oligodendrocytes and neurons, and now, it's a different distribution. So when you look at the astrocyte markers, right, they're over here, which is where they should be. It's the oligodendrocyte canonical markers in blue, and those are the ones that have the highest z-scores. And now if you look at the neuronal distribution, you can see it's in black over here. Those are the canonical neuronal markers, whereas the oligodendrocyte and astrocyte markers are over here, okay? So this is really fun, because when you start doing it and seeing how robust the patterns are across all of these independent data sets produced using all kinds of different technology platforms, what you realize is that, there's no other way to be analyzing these data, in my opinion. Another thing I like to point out is, if you take the genes, like the top 20 genes based upon their predicted expression specificity for astrocytes, and you go to PubMed and you search for the gene symbol plus the term astrocyte, and you ask how many studies come up, you can see, like here, this is a BDNF receptor. There's a lot for that one, right? But then you get down to number 6, 0, number 8, 0, number 9, 0, right? So same thing for the other cell types, right? So what are the odds that these genes aren't doing something important for the biology of these cell types? I think they're quite low, right? So if you're interested in the biology of this cell type, this kind of information is really a gold mine. In practice, again, what this means is that for many groups of genes, it doesn't matter where in the brain you look, and it doesn't matter how you look, they are consistently co-expressed, right? So here are the top 10 genes from that consensus astrocyte signature, and these are 4 independent data sets from adult human brain produced using different technology platforms, so 3 different kinds of microarrays or RNA-seq, and looking in 4 different brain regions, and you can see it doesn't matter where you look and it doesn't matter how you look, they're consistently co-expressed. Same thing down here. These are the top 10 oligodendrocyte genes, the top 10 neuronal genes, et cetera, right? So we've been kind of looking at this, again, very systematically. Lest you think all we find are signatures of just the 4 major cell classes, I'll show you a few other examples. This is sort of a dated and partial list of cell types for which we've identified signatures so far using this strategy. I think I showed this earlier. This is just, I have to pick examples that have, very obvious in situ hybridization patterns that are cell type specific. So choroid plexus is always one of them. I showed this earlier. Here's the top 3 genes in a choroid plexus module in an infant human brain data set, and they're in situ hybridization patterns in the mouse brain. You can see them all lighting up the choroid plexus. Here's another example. This is a signature of medium spiny neurons, which is the major cell type in the striatum. I'm going to highlight these 3 and just show you the expression patterns in the mouse brain atlas, which are right there in the striatum. Now, why did I not show the first, which is the one I should show? Well, because that's what it looks like in the mouse brain atlas, right? So now the question is that a probe failure or is it a species difference? And I don't have the answer, right? But that's something that my lab is working on because we've now been collecting mouse expression data. Believe it or not, it's one of these rare cases where the human situation is far superior to the mouse situation. There's a lot more human brain gene expression data from non-pathological samples than there is mouse brain gene expression data, right? So it's easier to do these kinds of analyses in the human. But this could be an example of a bona fide expression difference between the species and a particular cell type. Here's another example, Purkinje neurons. This is kind of an, yes, question?
In this case, you start with the previously published gene set to annotate your networks?
In some cases, when available, yeah. Sometimes we just know.
How do you know that a particular module represents astrocyte without prior information?
Right, yeah. So we use that as the hook, right? So like if there have been previously published studies that claim to profile a particular homogeneous population of cells, we can take that and then we can use it to look at our human data for the signature of that cell type, right? What that gives us, though, is genes that are specifically expressed by human astrocytes, right? Whereas all of the experimental data has been generated in model organisms, right? So that's one main thing. But there are cases where we don't have that information at all. And what you may know is that there's a handful of markers that are commonly used for this particular cell type. And it's not enough to do a meaningful enrichment analysis. But when you see that all of those markers have extremely high KME values for a particular module, then you would try and prove it histologically, right? To see that it is in fact specific for that cell type.
But an automated way of just a module overlap across the data sets tells you what these modules might be, but how do you mark it back to cell type specificity?
Yeah, well, I think there's 2 ways. You either leverage existing data, right, in gene set enrichment analyses, or you do histology and prove it yourself. Yeah. Yeah, so we do that too, but that's low throughput, that's, So Purkinje neurons, I think, are an interesting example because, they represent less than 0.5% of all the cells in human cerebellum, right? Probably far less than 0.5%. And yet, this is a data set consisting of only 24 samples of human cerebellum, and we're able to find a signature for the cell type, right? Those are the Purkinje neurons right there. So this relates to a question I often get that I don't have a good answer for yet, which is how abundant does a particular cell type need to be in order to find it with a given number of samples? And that's a hard question, right? And I think part of the reason we're able to find this is because the transcriptome of Purkinje neurons is quite distinct from the transcriptome of granule neurons, which are excitatory. Purkinje neurons are inhibitory, and therefore it really pops out, right, in this, even with only 24 samples. But for other situations, like subtypes of interneurons in the cortex, it can be harder, right, to try and delineate those. So the question is, how many samples do you need to find a signature of a cell type that represents 0.5% of cells in the cortex if its transcriptome is maybe not as distinct as the Purkinje neuron is from the background? And that is, I don't have a good answer for that yet. It's still work in progress. Okay, so lest I again give the impression that every module relates to a cell type, I'll just say that this is really the tip of the iceberg. We see many, many more things that we can't explain, including things that are quite robust and quite reproducible, and those are the things that I think are going to be fun to try and figure out. We also see things that we do understand but aren't obviously cell type specific, right? So here's a histone module. You can see these are all histone genes. I don't even know which brain region this is, but there they are. Here's a ribosomal module. This is a mitochondrial module. You can see many of these genes, in fact, almost all of them are members of the electron transport chain. This is a mysterious zinc finger transcription factor module. I have no idea what any of these genes do, and I don't think anybody does. Here's a module corresponding to gender differences. Here's a module that looks like it might have something to do with a phenomenon called LTP, which is long-term potentiation. Some people think that this is the way that neurons store memories. And basically, these are all immediate early genes that are transcribed in response to neural activity. This is early growth response 1, early growth response 2, et cetera, ARC, BDNF, et cetera. And we see this module in mice as well. To sum up part 1, I think it's quite clear to me that looking at the data in this way is just the way to go. You see the same things over and over again, and that's biology talking to you, So you kind of ignore it at your peril. And the reduction in data complexity is just the practical benefit of it all, right? It's much easier to try and understand the brain in terms of 50 co-expression modules than, several hundred billion cells and, yeah, cell type specific information. I mean, I should say again, it seems obvious probably to all of you in the room, but when I was starting my PhD, it was really kind of heresy, to argue that you could recover the cell type specific information from heterogeneous tissue samples like this. But I think, and this has now been demonstrated in many other biological systems too, that these cell type specific signatures are among the most robust and reproducible modules that you find over and over again. So when you're analyzing your data sets, if you are starting with a heterogeneous tissue sample, that would be the first question I would ask myself. Are any of my modules cell type specific, and how would I go about proving that? Yeah, and I guess the other point that is worth making is just that, these co-expression relationships, many of them are stable enough across many independent data sets that they give you a new quantitative framework for approaching disease or approaching species comparisons or really any kind of comparison, right? Because I can predict what a lot of expression patterns should look like in a de novo data set because I know which genes they're going to be co-expressed with. But if that prediction is wrong in a cohort of patients with schizophrenia, that might have something to do with the biology of that condition, right? Okay. Any questions about part 1? Yeah.
So, one naive inference from what you just said would be that mitochondrial or any other organelle's signature would be expressed in every cell type. Is that the case?
Yeah, it is. But are they expressed at the same levels in every cell type? No. So mitochondrial genes are expressed at highest levels in neurons, right? So you have to consider that, right? There's going to be, that's an area where I think we have a lot to learn. In fact, some of the single cell studies coming out now are claiming that neurons in general have much more transcript than glial cells, right? So I don't think anybody's really controlled for that yet. Yeah.
I was wondering if you can spend maybe 5 minutes just explaining a bit more detail how you go about doing the cell type specific profiling of the data sets.
How we do the cell type specific profiling?
Yeah, because I know earlier you referred to the VARIS database.
Yeah.
And that's one way of doing it. If you can just elaborate a bit more on how to do that.
Yeah, so I mean, as I mentioned in the last talk, we're collectors of gene sets, particularly neurobiologically oriented gene sets. So the data from Ben Barres' lab in the Cahoy et al. Paper in 2008 provide one source for a few different gene sets, right? What they did is they purified homogeneous populations of astrocytes, neurons, oligodendrocytes, microglia, et cetera. Maybe it wasn't microglia, but, and then they did a differential expression analysis, right? So astrocytes versus all other cell types, and they rank all genes based upon, some combination of fold change and p-value, and they publish that. So we take that list and we decide for ourselves where we want to cut it off, and we try to be conservative so that it's a relatively small set of high confidence genes that are expressed much, much higher in astrocytes than the other cell types. And then we build our co-expression networks in an unsupervised way, and then we ask which module, if any, out of all of the ones we find is significantly enriched with that set of genes, except we have it for 20,000 sets of genes, right? And not 20,000 sets of astrocyte genes or whatever. The cell type specific list is much smaller. It's probably, 100 or less. Yeah.
Yeah, because I ran into a similar problem that you have neurons versus astrocytes, right? And then there's only 5 genes that show that there's significant expression of other cell types. Like, for example, alpha-4 is going to be 35 times higher enriched in astrocytes compared to neurons, but that's one gene.
Yeah.
And the rest of them are maybe twofold.
No,
Well,
yeah, I mean, I think, I guess another thing to consider is, there's multiple sources now for these major cell types, I mean, like the Nat Hines study in Cell using the trap method is another source that we use. The Allen Institute has published lists on this as well. So I would try and collect multiple lists for a given cell type and then look at them all simultaneously, and they should show concordance. Yes.
I have 2 comments. You might be thinking on the same lines or something. For you're using an automated approach or looking at consistent pattern for cell type specific markers, right? Can you also use the similar approach for pathway or ontology based analysis, like looking at consistent patterns of set of genes in human brain for a specific pathway, like an immunological pathway?
Yeah. Yeah, no, absolutely. I mean, so like the molecular signatures database that I showed earlier incorporates gene ontology and KEGG pathways and biocarta pathways. It's got 10,000.
I was talking about like curating, because most of those pathway curations are based on or from the preliminary analysis in the literature from different tissues, not just brain specific.
Yes.
So I was wondering, is there a curated database for these ontological pathways which are brain specific?
Oh. Not that I'm aware of, but if there is, I'd love to know about it. Yeah, it would be great, right? I mean, that's kind of one of the problems with gene ontology in general, is that it's an abstraction to some extent based upon an integration of systems that never physically interact, right? So to the extent that you have something that's grounded in your biological system of interest, it should be more relevant, right, in principle. Yeah.
How different are they? Like, are they different between a liver tissue and a brain tissue?
I honestly don't know if any, I mean, like, you can ask about like ribosomes, right? I mean, and this is just by way of example, ribosomes were considered for many years to be invariant, right? Because how could a cell do without, just consistently functioning, never, erring translational machinery? And then work from Maria Barna, who was at UCSF, showed that actually there are particular genes corresponding to particular ribosomal subunits that are expressed in a tissue-specific fashion during development and confer specificity of translation on particular messages, right? So in other words, a ribosome is not a ribosome is not a ribosome, So yeah, I mean, that could also be regulated, for other organelles as well, in my mind. Yeah.
Rob had a question. Yeah, you had a question, I think, right?
Oh, sorry.
So, to what extent is this dependent on the particularities of the clustering? So, we have great methods for clustering, but they
Yeah.
to some extent.
Right.
So have you thought about going the other way? So let's say you have a Go category has 100 genes, and you say, okay, are these genes co-expressed above the level of random groups of 100 genes, even though you might not detect them as a cluster?
Yeah, so that question, if you guys didn't catch it, was, just given the fact that despite our best efforts, a dendrogram can vary, depending upon the parameters you use to build the network. What about taking a more focused approach where you're interested in this particular gene ontology category, so you're going to take that set of genes and then just ask directly, what is the similarity among those genes like in a given data set, and how does that compare to expectation? I think that's something one could do, and it could make a lot of sense if you were focused on a particular cell type or biological process. I think the way we approach it is that, I'm not going to claim this is optimal either, but we typically build a lot of co-expression networks for a given data set. And when we're looking for a cell type specific module, we'll try and find the module out of all of the networks that we build that shows the most significant enrichment for that particular gene set, right? Because the idea is that every one of these modules is an approximation of biological reality, and I don't know why one would be better than the other, right? So why should I choose? I'd rather let the people choose.
So you cluster the data at different thresholds?
Yeah.
Mike, how do you database these wonderful results? So Peter and I were talking about, you have a data set, you clean it up, it takes a day, then you do your WGCNA analysis. If you're lucky, now you end up with your neuron paper a year and a half after they've tortured you.
Right.
And then you'll end up with a short list. And I remember doing this with William Yang's lovely analysis of D1 and D2 paper, right? He's got a paper in neuron, there's a list about yay long, you take out the D1 and D2 lists, and you try to build around that.
Yeah.
How do we database these results? Where do they go?
I have a very complicated file structure in the computer in my office. Right. Yeah, no,
It's a tragic.
it kills me. It's actually one of my major objectives for the next 5 years is to build that database and integrate all of our results in a way that's accessible to everyone in this room and not just my lab.
Have you talked to Paul Pavlidis?
I have not, but I'm aware.
Definitely should.
Yeah,
So
no.
it basically cleans up all the geo stuff and it puts it in. You're cleaning it up at a different level than he does. So it would be great just to maybe just talk to Paul and throw it at him.
Yeah. Well, he's got that GEMMA database. Is that what you're talking about?
Yeah.
Yeah. No, I know. I mean, I feel like we're halfway there because we're doing things in a very stereotyped fashion, But yeah, that's something that's really lacking, I think, in general. All of these data sets are collecting dust in Geo and Array Express, and there's just no integration.
Okay, let's move to part 2, and
Okay,
then
I'll
we
just-
have discussion later. Remember, we have 1 hour of wine and cheese.
Okay.
So we can ask you.
Okay, fair enough. Okay, yeah. So the second part of the talk, I'm going to focus on prenatal development, and I'm going to talk specifically about a study that was just published, and this is a collaboration with my neighbor, or I should say my recent ex-neighbor, and not because anything bad happened to him, but because I moved in the stem cell building, and this was a study in which we were focusing on radial glia, which are the stem cells of the developing brain, and trying to identify a transcriptional signature of that cell type and compare it between humans and mice. So just a brief background on cortical development. And I'll start with the rodent schematic on the left, right? So basically, as your cortex develops, the progenitor cells are called radial glia. They're also called neural stem cells. And they live down in this area called the ventricular zone, right, which is near the ventricles of the brain, which are the fluid-filled cavities of the brain. And they produce neurons and then subsequently glial cells, like astrocytes and oligodendrocytes. And the neurons migrate along these scaffold fibers, which are actually part of the radial glia cells themselves. That's why they're called radial glia, because they extend this process from the ventricular surface to the pial surface of the developing brain. And the neurons migrate along that fiber and the cortex grows in this inside first, outside last fashion. So each new layer is built on the last, right? And there's 6 layers total. In humans, the structure is generally preserved, but work from Arnold's lab has shown that there is this proliferative zone, which is not entirely absent in mouse, but much, much, much smaller, which is called the outer subventricular zone. And that's also populated by cells that seem to walk like radioglia and talk like radioglia, despite the fact that they don't live at the ventricular surface. And so there's some who think that it's the existence of this zone and these cells within them that may be responsible in part for the difference in size between a human brain and the brains of other species. I was kind of more interested in a more general question, right, which is that, you know, when I started my lab at UCSF in 2010, the transcriptional profile of this cell type was unknown, right? And so I thought I should be able to find that. And if I can find it in humans, I can find it in mice, and I can compare them and see what's conserved and what's different, right? And maybe will lead to some understanding about what's happening within those cells that could possibly affect proliferation and the size of the cortex. So, you know, the broad hypothesis is that there could have been changes in gene expression in this cell type that might underlie neurodevelopmental differences between humans and mice, and potentially between humans and other species as well, right? So the problem that I was facing is that you want to have a lot of samples to do co-expression analysis. This was in 2010, so it was before the Allen Institute's data came out from fetal human brain samples, before the BrainSpan data set came out. And we were fortunate at UCSF because we have access to, you know, fetal human brain samples. But I didn't want to sit around for years trying to collect enough samples to do a meaningful co-expression analysis. So I had this idea, and the idea is we call GCAS, which stands for Gene Co-Expression Analysis of Serial Sections. And I've alluded to this a couple times, but the basic idea is that if you have one tissue specimen and you simply cut serial sections from one end to the other, and you extract RNA from every section, everything I told you about variation in cellular proportions will still apply, right? What are the odds you're going to have exactly the same number of radial glia in every section? You know, 0. So if that varies, then it should in principle drive the co-expression of genes that mark that cell type, right? At the same time, you control for differences between individuals, and instead of waiting years to collect 50 samples or 100 samples, you wait a few weeks to get your one, right? So we tested this, and this is the sample that we analyzed. This is a fragment of human fetal cortex from 14.5 gestational weeks. We cut almost 100 serial sections at 150 microns each from one end to the other. We extracted RNA from every section, and then we identified co-expression modules. We took, in parallel, a set of genes, which I lost it here. Here we go. No? Okay. So a postdoc in Arnold's lab had done an experiment in mice, and this is kind of the old-fashioned experiment in which he physically isolated mouse radial glial cells and other cell types in the developing cortex and did a differential expression analysis, right? So we had a set of genes that we thought would be expressed higher in radial glia than other cell types in mouse cortex, right? So this is from mice. So we use that as, you know, again, our hook in which to go fishing amongst our co-expression modules and look for evidence of enrichment. And we found 6 modules that showed significant evidence of enrichment with that set of genes, which suggested that these modules might consist of genes that are co-expressed in human radial glia during cortical development. So we calculated the module eigengenes. We calculated KME values. These are on a Illumina array, so we calculated KME values for all probes with respect to each of the 6 module eigengenes. We averaged those correlations for the 6, and then we converted that average into a z-score. And that's what this is showing you, right? That's the distribution genome-wide. So these in blue, these genes are canonical markers of radial glia, right? SOX2, PAX6, Vimentin, GLAST1, Notch, and they all have very high z-scores, right? Which means that they all had high KME values for all 6 of those modules, right? So that's good. Conversely, these are genes that are expressed in cells that have committed to become neurons, right? TBR1, FESF2, Relin, SATB2, and those were anti-correlated, right? So that was good, right? So in red, I'm showing you 4 genes that statistically are indistinguishable from the canonical markers of radial glia, but in the literature have not been described as being expressed by radial glia at all, right? So if this analysis is worth anything, it needs to be able to make predictions. So this analysis predicts that these genes should be excellent markers of radial glia. So we performed in situ hybridization in independent human fetal cortex samples, and you can see that all 4 of these genes are, well, you can see they're blazing at the bottom, right? That's the ventricular zone. So that's where the majority of these cells reside. So this is independent validation that these genes are expressed in the pattern that we predicted. But more importantly, right, this is kind of, you know, I hate to say it, but to me it's kind of paradigm changing, because what it suggests is that, like right now, everybody's talking about single cell, you know, and the insinuation is that the only way you can really measure, you know, cell type specific expression patterns is by isolating one cell type at a time. And I think that's not true. It doesn't mean you can't do it, but it's not always necessary, right? This is a case where you don't have to physically isolate or purify a given cell type using some kind of marker, right? All you're doing is taking a tissue sample and cutting sections and letting the patterns sort themselves out. And in my lab, we've done this now on 10 different samples, and we've probably identified on the order of 12 to 15 cell type signatures using this strategy, including a malignant cell type signature in a brain tumor. So at the same time that all this was happening, though, you know, all these other data sets were coming online, all these other fetal human brain data sets were coming online. So I wanted to take advantage of that. So I assembled 4 additional human fetal cortex gene expression data sets that are from comparable ages. That's 10 to 18 gestational weeks. These were produced using Agilent microarrays or RNA-seq, and they had anywhere from like 24 to 179 samples. And basically and then for mice, I found these 3 data sets, and these are from E14 to E15, which is comparable to the ages we're looking at in humans in terms of cortical development. And I effectively built co-expression networks in each data set. And you can see these are really heterogeneous, right? I mean, you've got different technology platforms, you've got different sampling strategies, so laser microdissection, macrodissection, single cell analysis, FACS sorting. You know, there's a lot of heterogeneity there, right? But I used the same set of genes that were expressed in mouse radial glia to look for modules in each of these data sets that were significantly enriched, right? So putative radial glia co-expression modules, and found a module with very significant enrichment p-values in each data set, right? So to integrate all of that, this was kind of the workflow, right? We have a total of 5 data sets from human cortex and 3 from mouse. So the 5 from human include the one that we produce with our GCAS sectioning strategy, and then the 4 independent ones, and then the 3 mouse ones. We find the module with the most significant enrichment for our markers of radial glia, calculate the KME values in each data set, and then we converted those into percentile ranks and averaged them within each species, right? And that seemed to me to be a conservative way to approach it, just given the heterogeneity amongst these data sets. And what we were interested in doing was then comparing genes based upon their average percentile rank, which is the proxy for the expression specificity within radial glia, right? That's the idea. Does that make sense? Yeah? Okay. Okay. So here, what we're showing is the scatter plot is comparing all genes based upon their predicted expression specificity in radial glia for humans and in mice. And overall, we saw pretty good agreements. We did enrichment analyses with genes in this box and then with genes in this box. This one here was all enriched with neuronal gene sets. Those are the things that are anti-correlated with the radial glia. These guys up here, it was all proliferative radial glia-related gene sets and astrocyte-related gene sets, because radial glia transcriptomes look an awful lot like astrocyte transcriptomes. If you look at the sort of creme de la creme, this is a blow-up to the right of some of the genes occupying the 95th percentiles in both species. Those of you who are neuroscientists or studied brain development might recognize Vimentin and SOX2 and PAC6 are in there, as well as Notch and some other ones. There's this whole cluster of cell cycle genes down here, and then a bunch of other stuff nobody's ever heard of, right? Okay, so we were interested in trying to identify, genes that were predicted to have high specificity of expression in human but not mouse radioglia, so genes that would be like down here, right? That's what we were interested in. The problem is that when you think about it, right, why would a gene, or potentially, why would a gene have high expression specificity in human but not mouse radioglia? There's two explanations, right? One is that it's not expressed in mouse radioglia, right, which is kind of what we're interested in. The other is that it's expressed in some other cell type in the mouse. Maybe it's expressed in neurons, right? It could be expressed in neurons and radioglia. That gets less interesting and more confusing. So the way we dealt with that was to impose an absolute expression level criteria as well. So we required that a gene be not only have higher predicted expression specificity in human versus mouse radioglia, but a much higher absolute level of expression in human versus mouse radioglia. And so that is sort of highlighted here in this blue row, right? So that corresponds to this quadrant. These are genes that have higher specificity in human and higher absolute expression in human. And we figured that genes that have evolved to have this Boolean expression difference, on in human radioglia, off in mouse radioglia, would fall in that category. So we're plotting now all of the genes based upon the difference in the percentile rank between their predicted expression specificity in human versus mouse radioglia and the difference in the percentile rank of their absolute expression level between human and mouse. This is where we're focusing our attention, and that led us to a small number of genes that are circled that were very, very consistent across all of the independent data sets, one of which was this gene PDGFD. Now, PDGF is a family of growth factors, and there's a rich history of literature and brain development that have really showed how powerful an effect growth factors can have on brain development, right? But this growth factor had never been implicated in brain development in the literature, right? So that was interesting. And furthermore, the receptor for this particular ligand, PDGFR beta, was right here on this plot. So it wasn't one of the most high confidence predictions made by our analysis, but it still looked pretty good, right? So now you have a case where both the ligand and its receptor are predicted by this analysis to be specifically expressed by human but not mouse radioglia during cortical development, purely starting from co-expression, right? This is a way of visualizing that, where when you take the top 10 genes from the consensus human and mouse radioglia signature, so these are the 10 genes that have the highest KME for the radioglia module across all 8 human and mouse data sets, and you look at a heat map of their correlations, this is human, this is mouse, right? Everything is the same except for PDGFD, right? We've added PDGFD to the mix. It's indistinguishable from like SOX2 and PAX6 in human, but in mice, it's not correlated with those genes at all. Why? Well, this is in situ hybridization for that gene in developing human cortex. You can see PDGFD is blazing in the ventricular zone, and in mouse, it's not expressed at all, right? So it's not there. So the gene is in the mouse genome, but it's not expressed in mouse radioglia. Conversely, the receptor showed a moderate correlation to the optimal markers of radioglia in human and no correlation in mice. And this might be kind of tough to see, but when you zoom in, I mean, this gene is also expressed by pericytes, which is a vascular cell type. But in humans, the receptor is expressed in the dorsal cortex of the developing brain, whereas in mice, it's only expressed in the ventral part of the developing brain. So there's been a shift in the domain of expression. So, at the risk of bludgeoning you with too much functional data, I might just kind of go quickly through the next few experiments, but we wanted to test the functional consequence of having these genes expressed in radioglia. So we did 3 different experiments. The first is that we applied an inhibitor, actually several different inhibitors, of PDGFR beta signaling in slice cultures of human fetal cortex. And sorry, that's a little bit truncated at the top. But effectively, you can use BRDU to label the fraction of dividing cells in the slice. And when we applied the PDGFR beta inhibitor, what we saw was this huge reduction in the percentage of cells that incorporated BRDU that corresponded to radioglia, which are the ones labeled by SOX2, right? So in humans, you give the PDGFR beta inhibitor, and effectively what this is saying is that the proportion of radioglia is cut in half. Whereas in mice, when you do the same thing, nothing happens, right? And that's not just the result of an increase in cell death. The second experiment is we actually took the recombinant protein for the ligand and we injected it into the ventricles of the developing mouse brain to see if it caused an increase in the proportion of radial glia. And we did, in fact, see a modest but significant increase of about 10% in the proportion of radial glia in mice. And we also saw this is kind of neat, but this is showing the distribution of radial glia from the ventricular to the peel surface of the developing cortex, and what happens when you inject this growth factor. And there's this subtle but reproducible shift away from the ventricles, which is kind of what you see in human evolution, where there's this expanded progenitor population that inhabits the outer subventricular zone away from the ventricles. But it was kind of a modest effect, right? Like a 10% increase is not that great. But then again, if the receptor is not being expressed by the radial glia, what do you expect, right? How can the ligand really have an effect if the receptor is not there? So the last experiment was to try and short circuit that, and we electroporated a constitutively active form of the receptor into the developing mouse brain. So electroporation is this crazy technique where you basically do an injection of a plasmid and then use these little, electrical mitts almost to give a shock to the brain that causes the radial glial cells to take up the construct and express the gene. It's like magic. And this guy, Tom Nowakowski, is really good at it. So he did it. And we saw that if you just activate the signaling pathway by overexpressing an active form of the receptor in mouse radial glia, it doubled the proportion of radial glia, and it causes dramatic shift, which is in blue, away from the ventricles. So it dispersed the cells away from the ventricles, right? So all of this suggests that this signaling pathway is active in human but not mouse radial glia. It's required for them to progress through the cell cycle at their normal rate in humans. And if you turn it on in mice, you cause an increase in proliferation and a shift in the physical location of these cells, right? So, and all of that started with co-expression analysis, right? So with that, I will conclude the second part. Pretty much everything I just said, I guess. Again, this GCAS strategy, we're excited about it in my lab because it opens up new ways of thinking about designing gene expression studies, right? Like, let's say you're interested in trying to understand what's different in terms of gene co-expression relationships between two different biological systems, right? Right now, with this strategy, in principle, you can try and address that with only 2 samples, right? If you have one sample from condition A and one sample from condition B, and you cut serial sections through each, and you build a co-expression network in each, you can do a differential network analysis with only 2 samples. And that's a different way of thinking about things. So the references for this talk 3 are right here. And I just want to thank everyone at UCLA who helped, again, set the stage for my career, including Steve Horvath, above all others. Dan Geschwin, of course, is my mentor as well and had a seminal role in all of this. Peter has been a great collaborator. Jeanette, thank you for your help in organizing things and putting together a great conference. My lab members and also the collaborators in the Kriegstein lab. Thank you all for listening for 3 hours.